Welcome back, everyone. I will now talk about two topics, and I chose them in response to some comments in the audience. I've noticed that many of you work on DNA methylation data, and I just wanted to give you a few slides that may interest you. And then I will talk also about a statistically important topic and also biologically important topic. When is it important to look at hub genes as opposed to just a standard differential expression analysis? So the topic I want to start with are to investigate aging effects on DNA methylation modules in both human brain and blood tissue. So here we applied WGCNA to multiple methylation data, and the goal was to find age-related consensus modules, basically co-methylation modules that are present in whole blood and brain tissue. Now, in principle, this is a very daunting hypothesis that you actually do find modules that are present in both tissues. Why is that? Because brain tissue is so different from blood tissue, you know. And but in any event, it turns out that age has a huge effect on methylation levels, and therefore this is a productive enterprise. So the analysis steps of this consensus module analysis proceeded as follows. First, we constructed signed weighted correlation networks in multiple independent data sets that I will describe in a second. Then we used our usual tools for identifying consensus modules, and then we related these modules to chronological age and gene ontology information. So I used publicly available methylation data and also data generated by my collaborator Roel Ophoff. And these came from various studies. They involved both whole blood data, but also notice data set 4. These are human brain data from about 150 individuals from 4 brain regions, and these data were published by Gibbs et al. So we used the blockwise consensus modules R functions in the WGCNA R package. Remember, this is part of one of our tutorials. It was incredibly straightforward. It's one R command, you know. And one thing I do mention is that here we looked at the 27,000 CpG probes that overlap between the Illumina 27K array and the 450K array. But as I mentioned, you can certainly apply the blockwise consensus modules function also to these 450K data only, which would then restrict the analysis to 480,000 CpGs. So by now you've seen these types of outputs. This is a cluster dendrogram, which really represents the output of the consensus modules analysis. There was one consensus, one hierarchical cluster tree, but it is really based on these 10 different data sets, you know. And as you know, the first color band shows the module color, and the other color bands report age correlations. So for each CpG, you report how highly is it correlated with age in these respective data sets. And when you carefully look at it, there is one module, the green module, there's this branch that contains a lot of genes, not genes, CpGs, that have a positive correlation with age in these data sets. So this is a module that gets hypermethylated as we age, both in blood but also in brain tissue. And the brain regions are frontal cortex, temporal cortex, pons, and cerebellum. Now, this is what I said earlier. There's this green module of great interest to us. Now, to relate the modules to age, you can, of course, correlate the respective module eigengenes with age in these 10 different data sets. And this table reports correlations, and in brackets it reports the p-values. And look at the green module in the last row. You find that this green module eigengene has fairly high positive correlations, particularly in brain tissue, right? Frontal cortex, the correlation is 0.7, and so on. So these are significant correlations. And the relationship in the cerebellum samples was slightly less than that in the other brain regions. Now, what's the next step? So now you have this module that relates to age, and if you would like to know, well, what is known about the CPGs, you know? And so you need to do some sort of an enrichment study. And so to do that, you want to measure module membership. We need this module membership measure. And as you know, when you have a single data set, this is defined as KME, the correlation of the eigengene with the individual variable. But when you have 10 data sets, you get 10 measures of KME, and you need to aggregate them. And so Peter Langfelder wrote a function called consensus KME, which allows you to input the multi-expression data, right? This is a list of 10 data sets, and either the module labels. You would just say for each CPG, are you part of the green consensus module or blue, and so on. And then this function outputs various measures of consensus KME, meaning, in essence, weighted averages of KME. Yes, so this function outputs various measures that allow you to rank the CPGs. And again, our philosophy is give the user anything they could possibly ask for. And so one is just a straight or average of KME. Then there are weighted averages, weighted by the sample sizes, right? And then there are Z statistics, and then there are P values, and there are even Q values, everything, you know. But here it turned out, I think we just used the average KME, it didn't have too much of an effect. And so then you can ask, well, what is known about genes that co-locate with these age-related CPGs? And towards that end, we used Jeremy Miller's user list enrichment function. And why? Because we were interested in brain functions and blood. Remember, it's brain and blood tissue. And the user list enrichment R function has a lot of gene lists that relate to that. And so we found these themes: nervous system development, neuron differentiation. It's all very interesting because the modules were also based on blood, you know. So this is other output from the user list enrichment function. Remember, it contains lists from the literature. So, for example, it found that the module was enriched for genes that were upregulated in neurons, according to the study by Cahoy et al. And also it found that it was enriched for CA1 area-related neurons, and so on. The other thing one can ask is this module enriched with polycomb group protein targets? And why would one even ask that? Because Teschendorf et al. Had found in their independent study and earlier study that genes that are hypermethylated with age are significantly enriched with polycomb group target genes. So these polycomb group proteins play a crucial role for maintaining the stemness of a stem cell. They play an important role in development. And it's very interesting to find this connection to aging, you know. So one thing about that previous finding is that it provides us with a gold standard for judging different methods for finding age-related CPGs, you know. remember how we always like to compare methods using real data sets. And here we have a real data set, and we can judge different methods for finding age-related variables. So, so what would a statistician do if he or she is confronted with 10 different data sets and wants to find age-related variables? They would use a standard meta-analysis technique, right? They would, remember in a couple of lectures ago I talked about the meta-analysis R function. So you correlate each CPG with age in each of the 10 data sets. You get 10 p-values or 10 z-statistics, and you could use Stufer's method or a rank-based method. And then you would find, let's say, 300 genes that, CPGs that relate to age. But now you can ask, if you do such a standard analysis described in the left panel, are the resulting CPGs, do these CPGs co-locate with polycomb group target genes? And so you can calculate an enrichment score, basically a chi-square statistic. And so yes, all of these methods lead to show that these genes that are hypermethylated with age are enriched with polycomb group target genes. But here's the interesting thing. When you look at the right-hand side, we show the results of ranking these CPGs according to their consensus module membership. Remember the output of the consensus KMER function. And what we find is that when you look at module membership-based rankings, they consistently outperform those based on a standard analysis. And so this gives you a glimpse that, yes, sometimes when you focus on consensus modules and consensus module hub genes, you really amplify the biological signal. Yes?
Is it just taking like the 200 top KMEs from?
I forgot the threshold. But yes, it was something like take the, if it was, I would say the top 500 CPGs. But for each method, you know, why? Because the enrichment score highly depends on how many CPGs you look at. And so we put all of them on the same footing. I forgot the exact threshold. But I do tell you that it didn't depend on how many you chose. If you look at the top 100, the top 300, these results are consistent. Now, interestingly, one can also relate this KME measure to other sequence properties. For example, you can say, well, what's the relationship between KME and CPG island status? And then we found, yes, these age-related genes that get hypermethylated with age are typically in CPG islands. And we compared these types of analyses to a standard analysis. And again, we found that the KME measure is superior to just a meta-analysis statistic. There were several genes that were interesting to my collaborators. I won't elaborate it here. And this short application really served several purposes. One is to show that a consensus module analysis based on DNA methylation data from multiple tissues can be productive, with the emphasis on DNA methylation data. Because I actually had a student, a doctoral student, who tried to do an analogous study based on gene expression data. And she worked on it for a couple of years, and we just really couldn't find anything. If you want to find age-related co-expression modules based on multiple human tissues, you won't find anything, at least not with WGCNA. Why? Because we tried it, but maybe you are lucky. If you want to go into a dead end with your scientific career, try that. However, if you, I do think that DNA methylation data are very valuable if you go across tissues. And so many of you have talked to me about that topic, you know, and so some of you study psychiatric disorders based on blood, and a typical reviewer comment is, but blood is not the relevant tissue. And I agree with that. I think we're all nervous about it, but I would say with methylation data, at least you have a prayer, you know, based on these findings. But it's a hypothesis, you know. Yes. Yeah.
So, like, if you the picture of the color modules for the different CPG sites.
Yes.
Is it always the case that with methylation, there's so many of them that don't get classified? Like, there seems to be a really big gray band.
Yes. Well, several comments. First thing is, when you do consensus module analysis across 10 data sets, the more you add, the more stringent you are, you know. And so this really gives you the low-hanging fruits. That's the first comment. But the second comment is the following. Remember how I told you this is based on the actually 26,000 CPGs that overlap between the Illumina 450K and the 27K array. We also wanted to be super cautious, and what we did is we decided that modules that were not present on the 450K platform shouldn't be pursued. Why? Because the 450K platform is arguably the newer platform. We had more confidence in it. And so we said, well, if a module cannot be found in 450K data, maybe it represents an artifact. So we applied a further filter. So there are a few branches here, not many, but a few of them we took out. Not to bias the analysis, but just not to mislead people, because some people will download, of course, the module assignment, and I just don't want that they have a module that represents a 27K chip effect, you know. Yes. Yes, other questions?
Can you say something about that dominating branch?
Oh, yeah, this
relative
one.
to the green dye that's,
This one.
When you see something like that, what does it tell you?
Yes, well, okay. Notice there's one extremely well-defined module. And look at, there's a color band that encodes X chromosomal genes. Because remember, so the CPGs on the X chromosome from women are more highly methylated. It's a mechanism to suppress, to facilitate X chromosomal inactivation. So yes. So if you have methylation data that contain both men and women, you will find one really distinct module. Yeah. Other questions? Yes?
You showed a correlation matrix that kind of looked like a heat map.
I don't remember. Oh yeah, this kind of thing, yes?
So if you look at the WB Dutch controls.
Yes, whole blood Dutch, yes.
Yeah, that column has a lot of correlations in it.
Yes.
Do you have any explanation for that?
Oh, okay, so WB Dutch controls actually is the age variable in that data set. So the columns correspond to age. And I gather then that age in this particular data set was certainly highly correlated with the green module, 0.4, but it actually correlated also with some of the other modules to a lesser extent. I mean, 0.2, it's not a very strong correlation, you know. Whereas there are other data sets where we find much less of a correlation, for example, type 1 diabetes data, you know. And the thing to remember is when you calculate a correlation, it very much depends on the range of the values. So let's say you start with old people, well, that's maybe the wrong example, but let's say people who are all in their 60s and 70s, there's barely any range, and therefore whatever correlation you can measure will be relatively small. Whereas in another data set, for example, these Dutch data from Roel Ophoff, they contain people who were in their 20s and contain people in their 70s. And so it just gave you much more power to detect a strong correlation. Other questions? Yeah.